Okay, welcome to Pavitra classes. Today you are studying about the biosensors. So, what are biosensors? A biosensor is an analytical device which converts a biological response into the electric signal. So, biological response. What is the biological response? It may be enzymes, hormones, antibodies, or nucleic acid, organelles, or whole blood cells. So, the simple thing is whatever the biological samples or biological response, biological material in the animals or in the plants, it may be converts to the electric signal. So the biosensor is analyzes the biological signals into the analytical signals. A sensor can be a single carbon electrodes, ion sensitive electrodes. It contains electrodes because it converts the signal into electrical signals, biological material. It may be the carbon electrode, ion sensitive electrode, photocell, an oxygen electrode, or a thermistor. The Professor Leyland C. Clark is the father of the biosensor. He discovered the biosensor. So, a good biosensor, what are the properties of the good biosensors? So, it should provide accurate, precise, reproducible results. So, it should provide a good result. It should be free from the electrical noise, and it should be cheap, small, portable, and capable of being used by the semi-skilled operator. So, it should be very simple to operate any semi-skilled or any less skilled operator. It should be cheap, small, and it should be portable. The reaction should be independent from the physical parameters. So, it should not alternate any physical parameters like pH, stirring, and the temperatures. This is the a good biosensor properties. How the biosensor should be for the users. So, next is the working of the biosensor. So, how the biosensor will work? So, the preferred biological material like enzyme, hormones, anything is preferred for the conventional method like physical or membrane entrapment. So, whatever the biological material we are taking, it will be first entrapped with the physical. Conventional method will be physical membrane entrapment, non-covalent, and the covalent binding entrapment. The preferred biological material, then it will be contacted with the transducers. It may be any electrical transducer, carbon electrode having ion sensitive electrode, photocell, or the oxygen electrode. It is contacted with the transducer to produce a bond analyte with the analyte binds to the biological material. So, whenever the transducer attaches to the biological material, it produces the electrical responses. So, by that electrical signal, the biological material is measured, and the analysis of the biological material, it may be enzyme, it may be hormone, it will be measured. The good biosensor example is whatever we are checking the blood glucose parameter in the diabetic patients. That is the one simple biosensor model of the thing. Then it is in some cases the analyte change to a product and have some probability to associate with the release of the heat, gases like oxygens, electrons, and the hydrogen ions. So the analyte change to the product. So after connecting to the transducers, in some cases it will be changed with its physical and the chemical properties like release of heat may be there, release of gases like oxygen, electrons, and the hydrogen ions may be there in the some of the biosensors. This is the picture of the biosensor. Here it is bioelement, whatever enzyme this is done. This is the analyte. This is the transducer. Here it give the signals. This is the electrodes. These two are the electrodes. This is a typical model of the biosensor. These are the parts of the biosensor. Next, coming to the principle of the biosensor. The principle of the biosensor is very simple. The biological material is immobilized, so it should not move in the one part to one part. The immobilization supports the permeable membrane. By entrapping that with the biomaterial, by entrapping with the permeable membrane, it is kept constant. Then the substance is measured to pass through the bioreceptors and the analyte. It will be converted to the analyte first. It is immobilized, then it is connected to the bioreceptors. Then molecular recognition will be done. After the molecular recognition, it is connected to the transducers. So here electrodes will be there. It will measure the signals, or it will convert the biological response to the electrical signals. Then the measurement will be done, and the data recording and the display will be there in that machine. In that data recording, the reading will be done. So parts of the bioreceptors: one is the bioreceptor, second one is the transducer, third one is the signal processor. So bioreceptor is the part where biologically derived materials, like enzymes, hormones, any blood particles, blood products, kept constant, and it is attached to the transducer. So when it is attached to the transducers, that is called as the bioreceptors. So biomimetic component that combines to the recognize the analyte of the interest. So when the biomaterials combines to the analyte or the transducer, that is called as the bioreceptor. Transducers are the when analyte is interacts with the bioreceptors, that changes the biological signals such as change in the temperature. Electrical change may occur, but it converts to the electrical signal. The transducer transforms these signal into the another signals, which is easily measured and quantified. So coming to the next part is the types of the bioreceptor. Based on the type of the transducers used, the bioreceptors are classified into 11 classifications, that is electrochemical biosensors, amperometric biosensors, blood glucose biosensors, potentiometric biosensors, conductometric biosensors, thermoelectric biosensors, optical biosensors, fiber optic lactate biosensors, optical biosensors for blood glucose, piezoelectric biosensors, and the immunobiosensors. So next we will study one by one. So electrochemical biosensors, first one is the simple device. It measures the measurement of the electronic current, ionic or conductance by the bioelectrodes. So here ion electrodes are used in the electrochemical, ion transducers are used for the ion sensitive electrodes are used for the electrochemical biosensors. This is the simple model of the electronic and the chemical mixed with the electronic behavior and the chemical behavior. The biological material will be converted into the electric signal. Second one is the amperometric biosensors. In these biosensors, usually the movement of the electron movements, electronic current determination as a reaction, the enzyme catalyzed redox reaction is taken in the amperometric reactions. Whenever the normal contact with the voltage passes through the electrodes to analyze the enzymatic reaction. So whenever the biological material is there, the enzymatic reactions produces the substrate or a product that can be transfer the electrons with the surface of the electrodes. So whatever the product is given, in that the electrodes will be measured. As a result, alternate current flow can be measured. So with the connection of the converted reaction, redox reaction converted to the product, whatever the biological material, that will give the alternate current flow. It can be measured with the amperometric biosensor. The substrate concentration is directly proportional to the magnitude of the current. So as the magnitude substrate concentration is increases, the magnitude of the current also increases. And the reduction of the oxygen is required through the oxygen electrodes. If the oxygen electrodes are used, reduction of the oxygen takes place. It is a simple way to form amperometric biosensors. The example is usually the determination of the glucose example with the glucose. The above description is about first generation of the amperometric, whatever the principle I told earlier, that is the first generation biosensors. Nowadays second generation biosensors are used. Amperometric biosensors are developed by in a mediator like the electron transfer to the electrode. So it contains second generation amperometric biosensors contains one more part, that is the mediator. Third is the blood glucose biosensors. It is, I told, it is throughout world it is used for the diabetic patients to check the glucose level of the blood. Blood glucose biosensors are the widely used throughout the world diabetic patients, and has a single use disposable electrode. So once use, it is one time use with the glucose oxide and the derivative of the mediator. So here ferrocin acts as a mediator in the blood glucose biosensors, and the shape of the blood glucose also it is taken place. Biosensors looks like a watch pen. Usually it looks like a watch pen. With the help of the hydrophilic mesh, electrodes are converted So here hydrophilic mesh is used for the electrodes, for the covering of the electrode. Blood glucose biosensors is a good example of the amperometric biosensors. So this is the second generation amperometric biosensors. Blood glucose biosensors are the widely used biosensors. Next is the potentiometric biosensor. In this type, changes the concentration. Usually ion concentration is measured in this. Ionic determination by the ions and selective electrodes are used. In this, pH electrodes are also used. Hence, a large amount of the enzymatic reaction is involved the release of the hydrogen ion. So, as it is ion selective, the reaction as the current passes, the hydrogen ions will be released. Ammonia selective and the carbon dioxide selective electrodes are the some important other electrodes which are used in the potentiometric biosensors. The released ions will pass the current and the measurement of the data will be taken place in the signal recorder. Next, potentiometric electrode and the reference electrodes can be measured with the help of the potential difference. It is directly proportional to the substrate concentration. So, how much the biological material is increases, as it increases, the potential difference also increases. The ion selective field effects transistors, lower price devices. It can be used to the miniaturization of the potentiometric biosensors. An example of the ISEFT biosensor. It is used for the open heart surgery. ISEFT. It is ion selective field effect transistor, which is highly used for the intramyocardial open heart surgery. This is also major biosensor used in the worldwide. ISFET for the heart surgery. Next is the conductometric biosensor. In the biological system, there are several reactions that change the ionic species. The electron conductivity can be measured with the help of anionic species. The example of the conductometric biosensor is the urea biosensor. To check the urea levels, urea glucose, urea, any urea particles, urine particles, these conductometric biosensors are used. Last is the thermometric biosensors. So there are many more biological reactions are connected with the heat. So whatever the reaction with the heat, it will be checked with the thermometric biosensors. Optical biosensors, these are utilized the principle of the optical measurements, usually in the like absorbance ratio. It is with the analytical part, fluorescence ratio, absorbance ratio, fiber optics. So here optoelectric transducers are used in this, and this will take the transducer elements. Biosensors involves in the enzymes, antibodies. Usually biosensors is not required with the reference sensors. Here reference sensors will not be there. Directly whatever the absorbance of the rays will be there, that will be taken, and the comparative signals are generated by using the sampling sensors in the optical biosensors. Next one is the fiber optic lactate biosensor. Usually in this, it is based on the oxygen concentration. Here oxygen electrodes are used, molecular, by identifying the effect of the oxygen in the fluorescent dye. Next is the optical biosensor for the blood glucose. So blood glucose is glucose oxidase enzyme is used by the blood glucose biosensor to break down the blood glucose. But it first oxidizes by using use of 2 electrons to reduce FAD to FADH2 enzyme, which in turn is oxidized by the electrode in a number of steps. The resulting current is a measure of the concentration of the glucose. In this case, electrode is the transducer and the enzyme is the bioreceptor. Piezoelectric biosensors. The principle of the piezoelectric biosensor is used in with the sound vibration. So by the acoustic it is also called as the acoustic biosensor. The basic of the biosensors are formed by the piezoelectric crystals, and by the sound change, trembling of the crystals, positive negative charge and the sound change, it is measured. By using the electronic device, we can measure the certain molecules of the crystals on the surface and alter the response frequencies using the crystals. So by usually these are used in the cocaine. These are also called as the cocaine biosensors. For cocaine gas, usually gas phase have been developed by the antibodies of the cocaine to the surface of the crystals that will be generated. This is the picture of the piezoelectric biosensors. Here non-specific antigens are attached, antibodies, antigen reaction. Next is the immunobiosensors. The immune biosensors works on the principle of the immunological specificity and mostly coupled with the measurement of the potentiometric biosensors. So immunobiosensors act with the addition of the potentiometric biosensors. Usually immunological specificity and the immunological, whatever the antigens and antibodies, they are measured with the immunobiosensors. Last part is the application of the biosensors. Usually these used in the various fields. Food analysis. For food analysis, biosensors are used, especially measuring in the fermentation of the products and determining in the freshness of the fishes in the marine fishes, and also used in the measure in the cholesterol levels in the milk products, especially butters, milks. These are used for the cholesterol, checking the cholesterol levels. Second is that these are used in the biomolecules and their interactions. Lipids, carbohydrates may be there. That biomolecules interaction within the body can be measured with the biosensors. In the field of drug development also used, and also used in the crime detection part. DNA analysis also it is used. Medical diagnosis, especially first glucose analyzer in diabetic patients is used as a biosensors. Abnormal toxic substances produced due to the infection can be measured with the biosensors. Environmental field of monitoring in pollution controls, usually quality control of the drinking water and detection of the pesticides in the water. In the agriculture field, pesticide contact with the water can also checked by the biosensors. Industrial process control can be done. Manufacturing of the pharmaceuticals and the replacement of the organs can be done with the help of the biosensors. In military services, these can detect the toxic gases and also chemical warfare agents which are used in the world wars. In the military service also, biosensors are used for the what type of the toxic gases are generated can also checked by the biosensors. These are the major applications in the different fields of the biosensors. So this is about the biosensors, a working principle, application, and the types of the biosensors and the applications.